Well, thank you very much, Sam. It's been a pleasure to be here, and I've enjoyed myself a lot today visiting many of your laboratories and many friends here. And thank you very much for inviting me to this lectureship. I've been interested for many years in the biochemistry and cellular basis of antigenicity, a field that has that changed dramatically in the last 2.5 decades. And it's on one aspect of this that I'm going to be talking about today. And what I want to spend a lot of time talking about the mission of antigens in autoimmune diabetes that we've been working on in the past 10 years or so, as we went in parallel to the studies on the basics of protein processing, which I'll review in number one very, very briefly and go over it in a general review way rather than actually giving a lot of specifics in here. The dogma that permeated immunobiology for 80 years or so, since its foundation in the 1880s, was that proteins were recognized prior to catabolism by the host. Proteins were recognized in their conformational state prior to any kind of catabolic processes by cells of the immune system. That changed dramatically in the 1980s when it became apparent that actually proteins that entered phagocytes were highly immunogenic. And at the time that the initial concept, the initial paradigm rose about recognition of proteins in their native conformation, the issues of T cells and B cells was really well appreciated. In fact, most antibodies are indeed to conformational determinants. But the issue that then became apparent, and this is what I'll sort of, let me go slowly here, review with you in this first sequence, is that actually proteins were recognized by the cell system as small peptides resulting by their catabolism by the antigen-presenting cells, the phagocytes, a number of cells derived from the monocytes and the B cells of the host. So the protein that we did a lot of the initial studies that and are still continuing is a small protein, and this will show you the basics of the entire system. It's a small protein of 129 amino acids. It's chicken lysozyme. It's a highly immunogenic protein, and that's why you study it, because you can get a good immune response to it. What we've been able to do in the lab by applying hard chemistry to T-cell biological findings is to identify every segment of the protein, which is outlined in here, that is actually recognized by the immune system. And all of these have been done in a biochemical way, and I'll show you very briefly some of the data. This major peptide that you have here in green is actually cut from the whole protein, actually placed on the class II molecules or histocompatibility molecules. And it is well known now that it is the peptide in these structural studies in yellow, okay, bound to the class II molecule, that the T cells will actually recognize. Cellographic studies by David Fremont, who has been my colleague in all of the structural work on this. And this is the classical picture of how this peptide is bound in here between two walls of alpha helices and a beta-pleated sheet platform. The peptide stretches from this molecule here. This peptide, by the way, is hidden inside the molecule. It's not recognized by antibody. But yet the molecule is open. This is a molecule, is a very tight globular protein with 4 disulfide bonds. So it's reduced, it's open, and this peptide is cut and then placed on this molecule. This is the side view of this peptide as it is bound to the MAC molecules. As you can see, there are side chains that fit in very well with some of these sites, MAC, so-called MAC anchoring sites, that allow the peptide to actually interact with. And the binding sites are conditioned to the MAC alleles, and this is the most polymorphic gene of the species. And depending on the particular MAC, some peptides may bind, some peptides may not bind. And I'll give you the example of it in the diabetic mouse in a moment. But as you can see, as the peptide stretches, there are side chains that are looking up which is what the T cell actually recognizes, red side chain that are sticking down, which conditions whether or not the affinity of, there is an affinity of the peptide for that MAC and the degree of antigenic stimulation. So from this study, we've been able to identify those 4 families of peptides that are selected, and here is the core that is binding in this situation in here, the combining site of 9 amino acid core that sticks to that combining site. And we can actually then isolate this peptide, make synthetic peptides, and do affinity binding. And as you can see, there's an enormous range of affinities in here, for 40 nanomolar to 5 micromolar. And this is the amount of peptide that is actually displayed on the MACs on the plasma membrane. And what I have not shown you here is the number of T cells that arise when you give these kinds of protein that gives rise to many different peptides. And of course, this is the so-called immunodominant one, has a higher affinity, and it's the highest representation on the plasma membrane. So these are the 4 peptides. From this, you can do a number of examinations on what is the biochemical basis of this binding. There's hard science in this, in the sense of the sequence of the peptide that allows for the binding to it. The cores are always accompanied by flanks that establish add to the binding affinity. And there's always in these peptides a main anchor. For this haplotype, an acidic residue at the P1 site would be the major reason for the anchoring, or brings in the major binding strength to these peptide interactions. There's also some other peptides that bind that actually, as you can see here, do not have acidic residues, and these are much weaker binding residues, much weaker binding peptides, and they're less expressed on the plasma membrane. You can ask the question, well, what happens with this one over here? And what happens is that you have, where am I? You have a number of residues that are unfavorable, called hindering residues of these markedly. So if you then take a lysozyme molecule that you mutate and you get rid of these arginine here, you will have an increase in the binding affinity, more peptide binding, as well as higher number of T cells to that particular determinant. So all of these kinds of biochemical studies, accompanied by mutations in the MAC molecules and in the protein, have allowed precise chemical rules to be established for the interactions of different segments of the protein with a given MAC protein. A lot of these studies has involved isolation of the peptide by mass spec with my colleague Michael Gross in the Department of Chemistry. And this is the dominant peptide that you can see in a whole chromatogram of peptides. This peptide comes up very, very strongly. It has this binding affinity. It dissociates very weakly, or has a very long half-life as a bound peptide. And it can occupy up to a very large % of the class 2 molecules on the plasma membrane. So one can go by combinations of mass spectrometry, analysis of these MAC molecules, and a number of biochemical and cellular studies to diagram out of a given protein what segments are actually being selected. And of course, those are the immunogenic ones that you want to identify in the context, say, of a microbial pathogen as a target for vaccine development. And the last comment that I'd like to make is that some of the peptides are also post-translationally modified. And that's an important component of genicity of these residues in here. For example, in this peptide that we're studying, there's a tyrosine here, the side view, the sticking up. And this tyrosine nitrate, the antigen-presenting cell is activated and is making peroxynitrate because of the interactions with interferon gamma and microbial products. So you'll have a nitrotyrosine in here. And the interesting point is that you have the nitrotyrosine, is that you can get T cells that are totally specific to the nitrotyrosine peptide. and they don't see the wild-type peptide. You can also see actually oxidations of tryptophans in here. And interestingly enough, some of the arginines are citrullinated, which is an important post-translation context of rheumatoid arthritis, where there is abundant antibodies to citrullinated epitopes. The relationship of these to disease have still to be examined and determined. So let me go then, after this very brief overview of these issues, to then discuss with you the studies that we have been doing in autoimmune diabetes. And these, actually, all studies that are going on in Lysozyme in parallel. As we went with one, we decided to enlarge the lab to see what was unknown. From Lysozyme, we knew all the components of the protein, all the segments of the protein. From going to the autoimmune diabetes, you're facing completely an unknown. What beta cell proteins and peptides are being presented to triggering what is a fascinating autoimmune disease? So in this disease, there is a major MAC genes that are influencing the development of the propensity for the disease. In the human is DQ8, the DQ8 alleles, and in the mouse is the G7 haplotype. And they select for peptides with very specific sequence motif. This is actually rather striking, the specificity of peptides that are selected by these diabetic propensity molecules. And I'll show you the data along these lines. Surprisingly, many beta cell peptides are presented by antigen-presenting cells in the pancreas. A large number of them. And surprisingly, perhaps too, there's a lot of cells that arise in this disease. It's actually surprising how many of these T cells are there. I'll mention this to you, and I'll also mention insulin being one of the dominant autoantigens that you find in disease. The model that we all use experimentally to study autoimmune diabetes are two. One is the spontaneous one, the NOD mouse model, which mimics very, very closely the human disease. Genetically and in the cellular reaction that takes place, and that's the emphasis on studying it. And then there's a model that many labs have used in which you plant antigen under the insulin promoter. You study the reaction. We've done this by, say, planting lysozyme under the insulin promoter and getting mice that then you can do the biochemical studies that I showed you at the beginning applied to a disease pathology. But I'll focus entirely today on the NOD mouse model, which has been used so extensively in the past 15 years or so to study autoimmune diabetes. It is a spontaneous disease. You don't have to inoculate anything here. 80% of your mice are diabetic by around 6 months. Diabetes starts 4 to 6 weeks. If you look at the islets of Langerhans, you start seeing infiltration by lymphocytes. Important trait is the genotype here of the MAC molecule, the IAG7 one, which has features which are identical to the human diabetic propensity molecule, DQ8. It's a T-cell disease. These T-cells infiltrate the islets starting at around 6 weeks of age. So the question is, what are these T-cells recognizing in beta cells, and how are they doing this? This is the kind of question that we've been facing. There are also antibodies to beta cells that arise very early in the disease. These antibodies have no effective function whatsoever. They do not cause any problem in diabetes. This is entirely a T-cell disease, and you know that because you can isolate these T-cells from islets. You can grow them, even though there are very few actually in absolute numbers that you can isolate from an islet. You can actually grow them selectively as clones of T-cells. You can transfer these then to mice that are of the same NOD that have the genes, but they have no lymphocytes because you've induced mutations in the recombination genes in RAGs and in the RAG genes, and then you can actually ask the question whether your T cell is actually doing something, the one you isolate. And again, the idea for us is to match peptides to T cells that are causing diabetes and see what the rules are for the development of disease in here. And if you like to look at slides, this is a mouse that received an inoculum of about 10 million diabetogenic T cells, and there's nothing happening in these islets. These are totally normal islets. By about 48 hours, 36 hours, the whole thing comes up. Now, you can see now lymphocytes starting to infiltrate and break the structure of the islet. You can see them around ducts. Some islets are still normal. There's a few lymphocytes coming in. You can see the infiltration here. 24 hours later, this is totally destroyed. The mouse is fully diabetic and hyperglycemic for life. And you can see now the complete islet being infiltrated here. You have neutrophils infiltrating, activated macrophages infiltrating, natural killer cells coming in. Of all of those, the one that is killing the islet is the activated macrophages. So the T cells migrate into that and start the inflammatory reaction. And what you've done in here by this experimental manipulation simply is pack the entire diabetic process that was taking 20 weeks or so to develop into 4 or 5 days. But that allows us experimentally then to manipulate the system and importantly to ask the questions, are these T cells that are matching a given beta cell peptide actually diabetogenic? And you can see the transfer of the disease. 100% of the mice will get diabetes in 3 to 4 days like I just showed you, depending on if you give 5 to 10 million cells. If you give less, it takes a little bit longer to actually get the diabetic process going on. The breakthrough in this field, I think, or I'm sure of it, came from the studies of Hugh McDavid several years ago, in which he related a specific MAC allele to actually the development of type 1 human diabetes. Okay. This is just to tell you the questions that we are asking. What are the features of the diabetic propensity MAC molecules? In other words, bring all the lysozyme story into this spontaneous autoimmune disease. What kinds of peptides are bound to G7? I'll call G7 the one in the mouse, which is the one that we mostly studied. Beta cell peptides are selected. And which beta cell peptides are actually recognized? Are those peptides presented in vivo? So this is the classical studies that McDavid published, Nature in 1987 with Todd and John Bell, in which actually they related particular DQ haplotypes to the development of insulin-dependent diabetes mellitus. And this was a major issue. I think this is the best study of the autoimmune diseases insofar as the relationship between MAC development and the actual disease process. Shortly thereafter, he showed that the NOD mouse had exactly the same problem in DQ that the DQ8 molecules had, and so this was a clue that this was in fact the features that actually gave the susceptibility to diabetes. The 3 mouse strains that develop the 3 species where you have development of spontaneous diabetes have exactly the same problem, and one critical amino acid. Okay, and as I'll show you here, this. We did the crystal structure of it with David Fremont in 2000. At the same time, the group at Scripps did it at the same time with a small peptide that was associated with diabetes. We did it with the lysozyme peptide. And the year later, Don Wiley actually showed it with the insulin peptide DQ8. And the structures were all very similar. And I'll show you the one that was published by David from our own studies. And the issues that these molecules have is that all MAC molecules at the end of the binding groove have an aspartic acid that forms a salt bridge with an arginine. From asp at beta 57 forms an ion pair with arginine and alpha 76. The diabetic propensity molecules are different, and they have a non-aspartic acid at that site. And that residue here leaves this arginine. Okay. You can see, and then this so-called P9 pocket, which this is the base of the P9 pocket, makes it very, very shallow and has a different composition right now. That single amino acid is the determinant. You can actually mutate the NOD so it goes back to wild-type aspartic acid in here, and those mice don't get diabetes or get the autoimmunity. So that is the key site that is responsible for the go, no-go for the autoimmune process. So the issue is if the MAC molecules, only raison d'etre is actually that they bind peptides. And here is this one here giving you that change. Your expectation was that the profile of peptides from one or the other, wild type or the NOD, the diabetic one, should be totally different. And in fact, that was the case. Actually, that study wasn't done until 3 or 4 years ago when we decided then to develop the analysis of them in a number of ways. And what I'm going to show you in the next 4 or 5 slides are studies on normal antigen-presenting cells. These are B lymphoma lines that express G7 or mutants of G7 or DQ8. And what they're doing is these cells are normally processing our proteins. They're normally processing, they're normally placing it on their MAC, the various peptides from the natural proteins of our natural processing of our proteins. So the question is asked, what is their biochemical composition? What do they show that actually it may be unique? And what I'll show you is a wild type line that contains the IAG7, which is made of an alpha chain of the D haplotype with DG7, which has beta 57 serine. And this is the key amino acid. The beta 56 also has his ser in the G7. And then we've mutated these to a pro-asp, which is the wild type on G7. And I'll show you, compare peptides here to there, because the difference is remarkable. And that is caused by these it is really the one critical amino acid in the beta 57. We've also made from the same from the same lymphoma line, we've transfected it with DQ8 molecules, as you can see here in this flow cytometry, and done the analysis of the peptides that I'll present to you in a moment. Okay. Interesting enough, antigen-presenting cells of DQ8 or AG7 can present to many T cells of the mouse. So the identity here allows for the recognition of many peptides presented by, as you show here in this, where you have islet beta cell peptides, an antigen-presenting cell or one or the other. This doesn't happen with all the peptides. It happens only with some. And this is the actual answer to the large series of studies that were done. So what is done in here, you grow these lines, you isolate around 10 to the 10th cells, you lysol your cells, you isolate the MAC molecules, you denature them completely, fly all the peptides, and then you take all this peptide gamete to your mass spectrometry kits, and you should be in good relationship with them, and they'll give you now sequence analysis of hundreds of peptides. It's an extraordinary technique. I'm impressed. And so now you get an analysis of hundreds of peptides, and you can examine what is the difference and similarities of all of them. So first of all, as a general features, peptides are selected for the class 2 molecule from vesicular proteins. This is normal catabolism of our own proteins. They're also selected from the cytosol too, and from endocyte protein. They always come in the class 2 system as families, which I'll tell you in a moment what a family is by giving you an example of it. Now, the feature here of the diabetes propensity molecules is that 80 to 90% of the peptides selected by GQ8 or G7 have acidic residues at the C-terminus. Okay. It's a highly specific selection of peptides. It's one of the most specific of the MAC molecules that have been recorded. And most of the selected peptides, interesting, contain more than one acidic residues at the C-terminus. Okay. And you can show by a number of studies that those acidic residues are now forming an ion pair with the unpaired arginine 76, which was unpaired because there was no aspartic acid on the beta chain to form the salt bridge. And studies that were done specifically with G7 lines, the peptides extracted from the mutant did not show the preference for acidic residues, again showing you that aspartic acid was the key one in there. And, peptides shown bound to one were not found bound to the other. We also made F1 lines of G7 with other haplotypes, and they're distinct peptides that go into one or the other. That was important to show because there's been issues in type 1 autoimmune diabetes about the homologosity of the DQ8 or the G7 molecule to develop disease. So the issues of competition on haplotypes has been raised, and this study clearly showed that it was not the case. Let me give you 2 or 3 examples of it. Here's a summary of all the different peptides from the G7 molecule, and this is the G7PD, the mutant. Pay attention to it here. Here are the acidic residues, aspartic and glutamic acid. You can see the incidence of it, close to 80% in these runs in here. Okay. While by taking out now and going to wild type, you've lost its specificity here. The rest of the sites, there are minor differences, but they're not particularly impressive in this selection. So the important point in here is the P9 pocket, okay? And you make the selection of this particular population of peptides. And with DQ8 it's exactly the same. It's glutamic acid and aspartic acid. And again, you have the high preference of them. With DQ8, you also have a presence for some acidics at the first binding site, at the P1 anchor site. And here's an example then of some of the peptides you actually isolate. Here's one. For example, here's the 9 amino acid core that had been defined in the Lysozyme system, as well as by other laboratories too, by the way. So here's the 9 residue core. But this is what I'm saying to you. The cuts, as proteases are cutting these peptides, the flanks are imperfect. What we think really happens is that the molecule unfolds, the MAC binds to it, and protects that site from catabolism, and then amino and carboxypeptidases then trim the peptide to the edge of the combining site. That trimming is imperfect and gives rise then to these flanks of different sizes, and I was telling you in the other previous studies how these flanks actually contribute to binding energy, and you can see they vary greatly. But here is the core. And this is what I was telling you about acidic residues at the C-terminus. Here's EED, for example. And what is in blue here is that this is the anchor at P9. Now you can ask me the question, you should, why do you know that this is the anchor at P9 and not this other glutamic acid or aspartic acid on the sides of it? And what you do then is you make synthetic peptides and do binding affinity reaction with purified IAG7 molecules. We get them purified from baculovirus preparations, and then you do binding affinity measurements. And this is the kind of experiment that tells you how you can identify the precise binding site of the peptide. And what you do is you place peptides, you make synthetic peptides in which you change each one of these residues for a residue that you know hinders the interaction, weakens the interaction. You put lysines in there. And if you see at this site, if you put a lysine in here, you don't change the binding affinity, which goes from 0.4 for this micromolar of this peptide to 0.3 micromolar. There's no change at all. You change this D over to K, there's no change whatsoever, but you change this one, which I told you that's the one that is the anchor, and now you change it completely. Okay, you weaken the interaction. So we know it's coming in through, this is the P9 site. And of course you can ask the question, why? And not going through the others. And the answer is that if you also put lysines at P4 and P6, you weaken the interaction. And had the AC gone to anchor this at P9, that would have made this lysine at P6 unfavorable. Had it gone through here, it would have made this lysine at P4 unfavorable. So the MAC then selects the peptide that fits in perfectly at MAC, and it's the constellation of all these MAC anchors that makes a good fit in here and makes this selection for this particular sets of peptides. So this is therefore the anchor site for this particular protein. And that has been defined for a number of them that were isolated from them. So that is the summary of what was the definitive studies establishing what is the features of peptides bind to the diabetic propensity molecule. That time, there had been a lot of discussion what went in. I think this, with hundreds of peptides, with this mutagenesis experiment, convincing established this is the way it goes. So now you have that information, and then you like to move it forward to what is a very difficult kind of approach, which is going on in the lab right now, and this is not definitive what I'm going to tell you about, which is we now find out what are your real autoimmune antigens now. The ones from beta cells that are doing the job. I don't care, you could say, about what are my natural ones. That allows you to establish peptide binding motifs, but now you should go to what really is transpiring here. And this is a difficult exercise for many reasons. Reason number one is you cannot get many beta cells. I can get many lymphocytes out of a mouse, but I can only get 300 islets, each of which has about 1,000 beta cells, and start doing your math. And if I need for mass spec at least 10 to the 9 cells, and of those cells that I get out of the islets, there are very few that bear class 2. This is a very logistically very difficult exercise. And then there's this issue here that you have to bear in mind, because this makes this interaction fascinating but very complex. And which is the last issue that I will raise today. And that is that beta cells do not express class 2 MAC molecules. So the immune system is blind to beta cells. The immune system is never going to recognize them. The only way that the immune system, the T-cell system, is going to recognize them is if the beta cell antigen are passed over to the presenting cell, to the macrophages or dendritic cells, and then present the beta cell antigen. So it's a cell and antigen transfer system. And that's very well documented, and now you see data on it. So beta cell antigens then need to be transferred to the APC, which then will process them, as you saw in the various examples that I gave you, the lysozyme. Okay. So there is this kind of situation that makes it kind of difficult then to do the biochemistry. Now the fascinating thing here is that islets of Langerhans of all species, normal or non-diabetic, actually contain APC within them. And these have diabetogenic peptides bound to their class 2 molecules. So in other words, monocytes from blood cross into islets of Langerhans. And within them, they actually plaster to the surface. We've seen this by intravital microscopy. And then they take up beta cell granules in high density. Those are the ones that are presenting. Eventually, those APC drain to the pancreatic node, and that does the priming of the diabetogenic T-cell. This is the cycle that we believe is taking place. So to obviate a lot of the issues in here, what we're doing right now, which is working, a lot of work still to be done, is we had to generate an antigen-presenting cell where we could get 10 to the 10 to do the biochemistry studies. And what we've done is generated insulinomas from SV40 transformed islets and transfected them or induced the expression of MAC molecules. So you have now an insulinoma that have MAC, and now you can isolate from them the MAC and see what beta cell proteins are presented, and in fact, what is their composite, what is where do they derive from, and what is their sequence. At the same time that you're doing it, because you want to match them, you isolate the T cells from islets. So you have now peptides isolated from one side, T cells isolated from the other, that you can transfer diabetes or not, and now you match them. And you examine then the reactivity of T cells in vitro as well as in vivo, and by transferring the T cells to rat mice. So this is, I'm not intending for you to read these, but there's a lot of beta cell proteins and peptides being presented. And they have this beautiful motif of acidic peptide residues here on what will be the binding to P9. Many of these, we've made synthetic peptides, and many of these we know that what we are telling you here, that this is the P9 anchor, is actually correct. Okay. By the way, with all of this data, we've made a very strong computational analysis of whether you can make prediction of what are peptides bound to G7. And it matches beautifully. We have a number of unknown proteins now that with this information from mass spec, we've been able now to make very good predictions of what are these peptides that bind. But here's a whole range of them that we find. We have T cells against many of them, secretogranin, carboxypeptidase A. These transfer diabetes. Many of them, by the way, are secretogranin here, chromogranin, synaptotacmin. Many CNS proteins derived from neurons and CNS cells are isolated also from these beta cells. So many of these have been isolated. Many of these actually cause or contribute to the diabetic process, which is rather fascinating and interesting. The plethora, the complete breakdown that you have of self-tolerance in this kind of an autoimmune disease, and the number of peptides that you can actually identify as immunogenic. Let me show you a couple of examples of this that we're still working on. For example, this was one of the clones of T cells that was isolated. And I show you this to match, to answer the question that the peptides from the insulinoma is a valid approach biologically. For this reason, you can isolate your T cells, and what you do is you take your isolate now beta cells from a normal mouse, and you bring in your T cells, and you see your T cell is firing numbers of beta cells and the IL-2 response in here on your vertical axis. You had to put in antigen-presenting cells in the reaction. Okay. So again, if you had no APCs here, this would be flat. So that means the beta cell proteins were in some way or another released. The APC captured them and now presented it to our T cells that we had isolated, and we had it as clones. And those same T cells that were reactive with normal beta cells are also reactive with the insulinomas that actually contain the IAG7. So they are exactly reactive to the same peptide here as it is in here, as expected, because there's T cell specificity in the reaction. Here's one clone against secretogranin, which gives you a very, very good reaction against actually the peptide that was isolated from beta cell, which we made now synthetic peptides too, and they're highly reactive to it. So one of the issues therefore that is very apparent, what I said to you a moment ago, was the enormous diversity of T cells and immunogenic material that is elicited in this autoimmune disease. Now, one of the proteins, when we transferred from to the computer, indeed this is sometimes distorted, and you can see it distorted here. But one of the T cells that are most abundant in islets of Langerhans are against insulin fragments. This was not discovered by us. This was discovered by the group of George Eisenbarth and Wegman in Denver. Made beautiful studies showing that there are a lot of cells against insulin there. Now, there's a number of interesting issues about these T cells to insulin. They may represent, if you take and you isolate T cells from islets and now culture them, you can find that about 1/10 or 1/3 of the T cells that you're isolated are against fragments of insulin. And the hot spot here is actually this segment of insulin from residues 9 to 23. Most of your T cells are direct to that. Okay. Now, the features of this is rather peculiar and rather interesting. By the way, insulin-reactive T cells do transfer diabetes. So this is a diabetogenic T cell, and here is one transfer, one of our own clones, and you can see the islets in here, but heavily infiltrated by lymphocytes in here. And this mouse is becoming diabetic. So there's been a lot of discussion where this is the dominant autoantigen. This is what starts the disease, and this is what drives the disease to completion. Okay? I have a reserved position in here. I think some of these are definitely diabetogenic, but I think there are other diabetogenic T cells coming into reaction. So there are also T cells against C-peptide, for example. They're also highly diabetogenic, as well as the ones that I showed you before from the biochemical analysis. But one of the features that, and now to finish in the last 3 or 4 slides, the talk that to me is amazing is what I have shown you here. And this is an issue that needs to be explained in the context of this autoimmune disease and as well as others, because this has also been seen in autoimmune encephalitis, what I'm describing to you. With Lysozyme, and that was the good thing of having this study model protein, high affinity peptide translated into high selection of peptide, high immunogenicity. A lot of T cells against that lysozyme peptide. That's what I wanted to show it to you about, the reason for it. But with insulin, here is now an interesting feature, because if you take a look at it, the insulin peptides bind to G7 with very low affinities, in the low micromolar range. They have a very fast dissociation rate. The dissociation rate is less than 2 hours. While the dissociation rate of lysozyme, I don't lose more than about 20% of the peptides in 7 days of incubation in vitro. It's extremely stable. Here it is a totally different story. And this is the major dominant autoantigen, seems to be major dominant autoantigen. Activation of the T cells actually requires about 1 micromolar peptide concentration. And the concentration of insulin in blood, I am told about, or in extracellular material, is about 1 to 2 nanomolar. So how can you get this? At this concentration and with very low affinity, and the concentration that you need is so high. The differential here is enormous. Okay? So this is an issue that I want to pursue, that we need to understand. How are these peptides presented in vivo? Okay? And I think the clue is the presence inside the islets of Langerhans of these antigen-presenting cells. And that's what I'm going to finish the talk about. So normal islets of Langerhans contain antigen-presenting cells from every normal mouse and humans. Actually, this is a mouse that its dendritic cells, CD11C, a marker of dendritic cells. A transgenic was done where there is the YFP molecule inserted in it. So all you have to do is look at these islets. They fluoresce. We're looking at this by 2-photon microscopy with Mark Miller, and you can see and count the numbers of these cells that are in the islets. These are the counts on normal mouse now. This is not even a diabetic propensity mouse. And each dot is an islet of Langerhans. And there, on the average, you can see them here. This is the distribution. Okay. Some islets contain a large numbers of them. Some islets contain 2, 3, and so forth. And this is the distribution of them. So this is a normal occurrence in all of them. We know the markers. We've studied very extensively. CD11C, CD11B, those of you who follow dendritic cell biology, and so forth, so forth. These cells actually were not described by us for the first time. Actually, my colleague and predecessor in the department, Paul Lacy, with Lafferty in Australia, had identified them in the 1970s. Okay. And Paul and Lafferty had talked about passenger leukocytes in the islets of Langerhans. Okay. And what Paul had done was, he thought that it is these cells that were the cause of the rejection of islets. He was isolating islets to do islet transplantation. Forget about diabetes. Okay? Because at that time, when he started working on it, the immunological basis of type 1 was not clear, was not known, as a matter of fact. But he wanted to transplant islets, and he knew that these cells were there. And he made a number of manipulations, which when I first heard it, I couldn't believe that these were true, but I respected Paul a lot. So I guess they were true, where he got rid of these cells, these very few cells that were there, and then he could transplant across allogeneic barriers with no immunosuppression. This is a very famous paper in Science where Paul and Joe Davey publish that they had been able to transplant allogeneic islets by just eliminating these cells without immunosuppression. So these cells are normally there. And what we've done now is very carefully study them and now place them in the context of this autoimmune disease. Okay. These cells If we isolate these cells right now, these cells are charged with peptide, okay, and will stimulate the immune reaction. We can take Islet, the insulin promoter bearing lysozyme. We can isolate these cells, give it to the T cells from the first part of the talk that I explained, and they'll fire beautifully. So these cells are normally being charged with beta cell peptide. And I think what is happening is that they're most likely taking insulin granules in very high content of the and having a very large content of the antigen right there in these cells. So even though you have very low affinity interactions, you're able enough to get sufficient peptide to start the reaction. I took this slide from Mathis Ben, who's been thinking about these issues very seriously for some time too. And what the slide is saying that the peripancreatic node is the place of sensitization, which takes from the pancreas, most likely from beta cells, and I think that's the answer, the antigens, and then migrates to the peripancreatic node. And that is the point where you sensitize T cells. We have been able, by a number of manipulations, to get rid of the peripancreatic node in NOD mice. And if you get rid of the peripancreatic node, you have absolutely no development of diabetes. You don't sensitize. So that site is totally required for the first development. So we believe that it is these intra-islet APCs, the ones that are actually doing the job of capturing antigen. And here is with two insulin reactive clones, 1.90 INS10, in work that was done by Matteo Levisetti in our lab. And you can see that if you take these T-cell hybridomas against insulin and react with islets that contain this APC, they'll fire very nicely. That interaction is blocked by an antibody to G7, showing the specificity of the T-cell interaction, but not by any relevant antibody. So there's definitely immunogenic antigen in these APCs. So the cycle that we are seeing right now is that these APCs come from blood monocytes, jump into the islets, stay there for a period of time, most likely will drain out into the peri-islet tissue to then go into the lymphatics and move into the node. And it is in this cycle while they are in the islets themselves where they capture this beta cell antigens or granules and then select for the peptide that I was telling you about. Obviously, the disease is a very complex disease. How you can actually escape tolerance to many self-antigen is still a mystery with this particular haplotype. I think it's related to the structure of the G7 molecule in itself that is responsible for it. But as you can see, it's fascinating in the plethora of T cells and the different specificities that generate. The goal would be at the end where you can say, fine, I have gotten this number of T cells with different specificities. What are the ones that are starting the process? What are the ones that are continuing the process? Which are the ones that arise after a certain degree of beta cell death? These are the questions that we want to ask. What is the potency of these various diabetogenic T cells? And that's what we would like to go in the next several years. So it's been a lot of fun then applying the basic biology of antigen presentation to this spontaneous autoimmune disease. I've been fortunate to have Anish Suri, who did a lot of these biochemicals. Michael Gross and Henry Rohr are in the Department of Chemistry, who did the mass spectrometry work. David Fremont has crystallized these molecules. These are my other colleagues and postdocs in the department, Boris Calderon and Matteo Levisetti. And this is part of our technical staff who did a lot of the binding and MAC isolation. Thank you very much again.
We have time for a couple of questions. There are
So the presentation of dendritic cells draining into a lymph node associated with the islets is fascinating. Is the NOD mice unique in terms of having that association, or is it common to have an associated draining node from the beta cell?
Well, they haven't been studied in many models, but it looks like that is the case. So, for example, experimentally, in one of the arthritis models, again, the draining node is the point of collection and sensitization, and that has been shown reasonably well. In the brain, it's also thought that the upper cervical nodes also contain some of the antigens in the presenting cell. This is one of the best examples that there is to it, because you can remove the nodes and you can actually take your lymphocytes, mark them with dyes and transfer them, and the only node that they will proliferate is the peripancreatic node. That is the only site where they go into activation. And you can actually, by the way, which I didn't mention, isolate the same dendritic cells that you've isolated from islets, isolated from the peripancreatic node, and they are charged with the peptide. So I think this is, to answer your question directly, I believe that there is more evidence besides this, but I believe this is a generalized phenomenon. The lymph node collects, and it's a site of sensitization.
George? You mentioned there were two models, the NOP model and the one model you were using. Are there any advantages to the second model that you described?
Yeah, the second one has certain advantages because you can plant in the beta cells your proteins in different amounts. So we have transgenes that express high levels of lysozyme, and so forth. And then you can ask the question, how does the amount there condition the development of diabetes? And you can then take T cells that are highly dominant and highly active and all those that are less and so forth. And you can then bring in quantitative aspect to the degree of injury that you're seeing. This is one of the advantages that I've seen in that model. And as you see, also you can use that then to take your APC out and then do the analysis where they're charged with antigen. Much more reagents in the Lysozyme work than in the NOD work, so you can use more reagents to actually track quantitatively the interaction.
And is there a mouse model, a couple of mouse models of type 2?
Yeah, there are models of type 2. Yeah.
So there's been considerable thought in years of ways tolerant prevent autoimmune diabetes, and there have been a number of schemes and some trials even.
Yeah.
Can you glean additional insight or strategy to where you think those sorts of approaches might go, given this very high resolution understanding of the process?
Yes, I think that's, the NOD mouse is well known, and people have criticized it, saying, well, it's not physiological because you can actually just bang your cage and your NOD mouse, you can control the development of diabetes kind of thing. Okay, so in other words, there are many different manipulations that one can do in the NOD mouse to stop what is a very complex chronic autoimmune disease. Some of them are what I would see as a strategy to follow is to use peptide therapy in a way. But peptide therapy at the beginning was used as, well, what I'm going to use the peptide is to block MAC, which didn't make sense that you could block MAC. So therefore, I'm going to stop your T cells to coming in. I think where peptide therapy is going is because involved in the development of this chronic autoimmunity, you have a lot of regulatory T cells that have same spectrum of specificities that these effector T cells have, which you can then bring up into the reaction by actually with peptide alone. So if you know exactly the epitope, you can target that kind of approach in a very rational way. There's been a lot of mistakes done with the NOD mouse. I mean, at the beginning, everything was glutamic acid decarboxylase. There are hundreds of peptides on glutamic acid decarboxylase. Glutamic acid decarboxylase, as far as we know right now, doesn't have such an effector reaction at all. And it was done in a way because the approach was a bit blind at the beginning. Let's just get a protein, let's just try and figure out what segments might do it. Okay? And I think it has led to different, into ways which are not physiological in that sense. I think getting that kind of of data, know exactly the sequence that is involved, okay, because you have the hard chemistry of it, can lead you to a more rational approach if you want to use peptide therapy, which I think is the way to go. Yeah.
Well, thank you very much for a very stimulating lecture. And I invite you to join us in the North Lobby for a reception.